Hello, my name is Natalia. I'm a second-year student at UC Berkeley.
My name is Tovar. I am an incoming freshman at NAU. We worked under the mentorship of Dr. Sao, the Chief of Vascular Surgery in the Department of Surgery. She specializes in the study of cardiovascular disease, specifically atherosclerosis.
Dr. Sao's lab mainly studies the role of resistin in the pathogenesis of atherosclerosis, especially during diabetes.
Atherosclerosis is a disease that begins with hyperlipidemia that progresses to fatty streaks in the arteries, becoming lesions alongside the accumulation of endothelial cells that promote inflammation and the buildup of fats, LDL, and other substances that accumulate in the arteries.
The progression of atherosclerosis is attributed by the adipokine known as resistin, that is resistant to insulin, that is produced in humans via macrophages and adipocytes in mice.
The purpose of our contribution was to study the expression of resistin in macrophages of humanized TG mice. We also studied the induction of resistin via LPS, an irritative stimuli that mediates arterial inflammation by activating the macrophages.
In addition, our project also aims to address the epidemiology of atherosclerosis and correspondingly cardiovascular disease, since there is a public health crisis specifically in first world countries in which diseases such as type 2 diabetes and obesity are most prevalent.
For our project, we used THP1 cells and transgene and knockout mice blood samples to view resistin expression. THP1 cells are monocyte-like cell line from peripheral blood of a childhood case of acute monocytic leukemia. The transgenic mouse produce human resistin via macrophages, whereas knockout mouse do not have the gene to produce resistin. The mice blood samples were gathered from in order to gather PBMCs that were later converted into the macrophages.
Thus, our THP cells and mice samples began as monocytes, or rather peripheral blood mononuclear cells, that were later converted into macrophages, which secrete resistin expression by using PMA/TPA. As you can see in this slide, is our day 1 THP1 of adding TPA/PMA. On day 2, our observations were that they began to further clump together and attach to the walls. In this slide, you can see the knockout mice sample after its first day of adding TPA/PMA. And by the second day, as you can see, there are drastic changes to the cell's shape, which also alluded to that they were ready to have LPS added to induce resistin expression. In this slide, you can also see the transgenic mice sample from day 1 of adding TPA/PMA versus day 2 observations in which the cells began to clump together and change shape.
Upon the monocytes' conversion into macrophages, each cellular sample was then given LPS based on different time variables. The first vial had LPS for 4 to 6 hours, the second for 24 hours, and the third one was left untreated to serve as our control group.
After our LPS treatment, we began the process of immunostaining our cells to later express fluorescence under the microscope using DAPI to express the nucleus of our cells as blue, as well as Texas red for resistin expression, and one vial with no dye, labeled as BF, or bright field.
As you can observe, the 24-hour sample showed a greater resistin expression. This is shown because the cells show an elongated shape. However, it is worth noting that our untreated THP1 samples also displayed some resistin expression despite the lack of LPS inducement. This conforms to the idea that resistin is still naturally produced in the body. Our experiments contributed to the lab by confirming that the utilization of humanized mice could serve as proper models, which then allows us to confirm that human resistin is in fact expressed in the macrophages.
Our final questions include, does resistin directly impact atherosclerosis, or does it just promote its development? Also, can TL4 receptors be blocked to treat resistin expression and decrease inflammation? As well as what causes the inner walls of the arteries to rupture? If HDL protects the body from cardiovascular disease, how could it protect the body from developing atherosclerosis?